Yes, welcome to Biology Made Easy. Today we are going to look at fungi still. Yes, so objectives for this lesson. Let's look at conditions that favor the growth of fungi. Fungi grow in warm and humid atmospheres. So you need warmth, you need humid, and you also need to have a decaying organic matter present. That's the conditions for fungal growth. Now, how do fungi land on structures? How do they get there onto the food? Now, they get there because of spores that are released. The spores of fungi are released into the air, and the air carries them, and they settle on structures. Now we want to see how they grow on structures. And this is a spore of a fungi, and this spore of Rhizopus, all right, I call it positive spore or positive strain. I'll explain later. So this is a spore. It lands on this food, which is bread, and the spore germinates into a hypha. The spore germinates into a hypha, and this is a spore case. The spore germinates into a hypha. The hypha grows and forms a great mass of hyphae. Okay? As you can see in the shot, the bread, you have bread there, nothing on it, but spores fell on them, and you see hyphae grown. So these are the hyphae grown. This great mass of hyphae you see in the bread, this is example of it. And all the hyphae form one mycelium. Now the hyphae is white, all right? With time, the hyphae grows black sporangia. So those black bits in the hyphae are spores, are sporangia or spores. And in the drawing, this is it. A hyphae grows a sporangium. The sporangium black bits, they contain spores, which are black. Now, when the sporangium matures and dries, it bursts open and releases the spores. So this sporangium has released its spores. The wind carries it, and it falls on other structures, bread, and it grows. These spores were produced asexually, and these spores will go and do asexual reproduction. We'll come to asexual reproduction. So this is how spores, fungi spores land on structures and they grow. Now, one thing to take note of is that this hyphae of these spores may be growing on the bread with other spores. So I can have hyphae growing, but this hyphae is not one mycelium. It may be growing with other hyphae. All right, other spores may have been released that are different from this one. That's why I call this plus strain, the yellow. It's a plus strain, one strain of hyphae. The same bread can have another strain of hyphae, and now I make it blue. And this one, I call it a minor strain, also growing. So if you have one substrate bread, it may not be only one mycelium. You have the yellow hypha grown from the yellow spore, and then you have another spore which is minus spore, and it grows another hyphae. Now, so note that the yellow spore, yellow strain came from a nucleus that was yellow. So if you have a hypha of a fungi, and there are so many nuclei in them, all the nuclei can be positive strain. You can also have another hypha, and all the nuclei will be negative strain. So if a positive strain and a negative strain are lying together, there will be sexual reproduction between the two. Hyphae with positive nuclei, hyphae with negative nuclei. You can have a few hyphae that has both positive nuclei, you have negative nuclei. Those ones are the ones that have undergone sexual reproduction. Let's look at the structure of Rhizopus. It's called a pin fungi. You see the hyphae are very thin, all right? Now, this is the upright hyphae called sporangiophore. There's a hyphae that lies on the bread. The substrate is called a stolon. That is what spreads the fungi to other places. That's the stolon. Then you have rhizoids. These are fungi inside the bread. So every fungi you see outside, it has part of its hyphae inside the structure. If the fungi is on the wood, part of its hyphae are in the wood. All right, these rhizoids are in the bread, and this another sporangiophore and sporangium. This part of the sporangium is called a columella. It supports the sporangium. So simply, this is a Rhizopus. So let's look at asexual reproduction in Rhizopus. Now you have a spore that has grown into a hypha. The upright hypha is a mature sporangiophore. The cytoplasm and the nuclei, they move to the tip. The tip is swollen, and the nuclei and cytoplasm, much of it fill the tip. It's going to form a sporangium, and very soon a wall is cut off. All right, and that wall is columella, and this part becomes a sporangium. The sporangium is filled with a lot of spores, and the spore is a nucleus with cytoplasm and a thick wall around it. And the sporangium matures, it dries, the sporangial wall breaks, it breaks with force. So the spores are released and scattered. The spores are light in weight, so they can be blown by the air or wind. They fall onto a suitable substrate, bread, and then it absorbs water, the spore break, and a hypha comes out. And the hypha grows and forms more hyphae are produced by the stolon, and very soon you have a mycelium. Plenty of hyphae. This is asexual reproduction. Yes, so you have a question on the asexual reproduction, okay? These are the questions. How is the columella different from the sporangium? That's one. 2, what is the function of the columella? 3, why are the spores dispersed by air or wind? Now let's move on to sexual reproduction in fungi, Rhizopus. Now sexual reproduction occurs during adverse conditions, dry conditions, or excessive heat conditions. Then sexual reproduction will take place to form zygospores that will ensure that the fungi's life is preserved during unfavorable conditions. Sexual reproduction. Now, we already said when there's a mycelium, fungi growing on, say, bread, it has a lot of hyphae, a lot of sporangia, but the hyphae are all structurally the same. They look the same, but functionally, they are plus strains and minus strains. Yellow strains, blue strains, but structurally, they look the same. Now, and we said that when the different strains of hyphae lie together, conjugation, sexual reproduction can take place. Sexual reproduction is by conjugation. And we explained what conjugation is in our last lessons on bacteria. All right? It's just parts of organisms coming together to exchange nuclei and then to produce individuals that are genetically different. Good. Now, so let's look at the process of sexual reproduction in rhizopus. You see these two hyphae, they are structurally the same, but they are functionally different. This is a plus strain, this is a minus strain. They are lying very close to each other. And so when that happens, short protrusions grow in their side branches are short protrusions grow. The short protrusions continue growing long, and the cytoplasm in the hyphae comes to fill the tips of the swellings. And this kind of structure is called a progametangium. Now, the swollen tips meet together and the wall cuts off parts of the progametangium from the tip. And when the wall cuts off, the part that is the tip is known as a gametangium. And the part that is suspending the tip, cut-off tip, is called a suspensor. So the tip gametangium, this part suspensor. Then this one too has a gametangium and a suspensor. Good. Now the next thing to happen is that the wall between these two gametangia dissolves. And when the wall dissolves, the plus nuclei and the minus nuclei, they mix, and then they pair up, and then they fuse. So fertilization takes place. Then, so when they have paired up and fused, a zygote is formed. But this zygote is multinucleate and it's a diploid zygote. Diploid. It has two of every kind of nucleus. You have one male nucleus, one female nucleus. And then it's a diploid. It's a multinucleate diploid zygote. Then the zygote grows a thick calcium oxalate wall around itself, and the whole structure becomes a zygospore. The thick oxalate calcium structure is to protect the zygote from losing plenty water. It's a protective case. So with time, the suspensors holding the zygospore shrinks, and the zygospore is released. And the zygospore is light in weight, so it's blown by the wind. So the zygospore is blown by the wind, it lands somewhere, and is dormant. It doesn't germinate. But when conditions become favorable, there's water, the zygospore will absorb water, moisture, and then the zygospore case will break, and a hypha will form. And notice that this hypha that grows, it has both positive and negative nuclei in it. All right? So this hypha is a reproductive hypha. That's how I call it. It has both positive and negative nuclei in it. So in this hypha, asexual reproduction takes place, and a sporangium is formed, and the sporangium contains spores, with the columella is still there. And the spores that are formed here, because asexual reproduction took place, there are positive spores and negative spores. And the sporangium matures, dries, and breaks to release the spores. And the spores that are released, some are positive spores, negative spores. And the positive spores, when they get the right conditions, moisture, warmth, good substrate, the spore will germinate and grow into a hypha, and then mycelium that are positive strain. Then the negative spores will also grow into hyphae that are negative strain. So this is sexual reproduction that occurs in Rhizopus. Sexual reproduction will take place to form zygospores that will ensure that the fungi's life is preserved during unfavorable conditions. Let's quickly look at nutrition in Rhizopus. Rhizopus is a saprophyte. Now, how does it feed? As we have said, this is a Rhizopus. The rhizoids are the root-like structures growing in the substrate. And what it does is that this fungus releases enzymes onto the food or substrate. It releases enzymes from its rhizoids. Enzymes will be released from its rhizoids onto the food or substrate, and the enzymes will digest the insoluble food substances to soluble forms, and then the soluble food substances will be absorbed into the fungus. So by releasing enzymes onto the food, or if a fungus grows on wood, by releasing enzymes onto the wood to dissolve it so that it can absorb the soluble products into its system, then it decays the food. It breaks down the food, or it breaks down the structure on which it's growing. So when we see fungi growing, much of it is growing inside the substrate in which it grows, and releases its enzymes into the substrate, dissolves it, digests the insoluble food into soluble products. Carbohydrates are digested into glucose, proteins are digested into amino acids, fats are digested into fatty acids and glycerol. Then it will absorb the digested food products into the whole system, by that dissolving. So now let's look at our assignment. What is the importance of feeding in the fungi, Rhizopus? We want to look at the economic importance of the feeding of the fungus, and then the ecological importance of the feeding of the fungi. You have a question on the asexual reproduction, okay? These are the questions. How is the columna different from the sporangium? That's one. 2, what is the function of the columna? 3, why are the spores dispersed by air or wind? Good. So the solution, how is the columna different from the sporangium? Columna is white in color, sporangium black. Columna is oval in shape, sporangium round in shape. Columna is light in weight, sporangium dense or heavy because it contains spores. It had a lot of nuclei and cytoplasm, so it's dense, it's heavy. Then the other question, what is the function of the columna? It supports the sporangium. Then you have another question, why are the spores dispersed by the wind? 1, they are light in weight, can be carried by the air. 2, they are thrown by the force generated by the splitting of the sporangial wall. 3, the sporangiophore carries the sporangium high up in the air. So we've had enough for the day. So we'll meet in our next lesson. Goodbye. Thank you.